In this video, we are going to discuss on the general characteristics of animals and an overview of their diversity. So I believe that it's with the animals that we can most relate to when it comes to learning and talking about living organisms. And that's because we ourselves are considered scientifically as animals. And it's very easy to distinguish for us, at least, what animals are. Well, at least for some types of animals. But I think you're aware that there are certain types of animals that resemble plants more than they do our stereotypical view of what animals are. So, we are going to summarize in several points the characteristics of animals, starting with their nutritional modes. So, all animals are considered as chemoheterotrophs, meaning to say we get our energy from chemicals, from breaking and making chemical bonds, and we also get our carbon sources from consuming organic materials. So we are chemoheterotrophs. So when we consume organic materials, unlike fungi, we actually ingest our food instead of just absorbing them. Number two, all animals are multicellular. So even the smallest ones, which are microscopic, like if you've heard of the animal, the tardigrades or the water bears, they are actually animals, but they rival single-celled eukaryotes in their small sizes. But they are still multicellular. Okay, so all animals are multicellular eukaryotes. And instead of their cell walls, or instead of their cells being rigid, because they're surrounded by cell walls of chitin or cellulose, animal cells don't have cell walls. Okay, there's an absence of cell walls, and instead, the structure of the animal body is kept in its integrity with the help of proteins like collagen. Then there's also a presence of unique types of tissues in animals that cannot be found in any other organisms, like the nervous tissue, okay, which allows us to respond actively to our environment, and the muscle tissue, which allows us to move, with the response that we have with our environments. Number three, animals' life cycle is dominated by the diploid stage. Okay, so most often the animal that we see is a diploid organism. The haploid portions of the life cycle are stored within that animal. For example, as a sperm or as an egg. So you compare that with the life cycles of fungi and plants, it's very different. Okay? And when fertilization happens, when the egg fertilizes the egg undergoes development first through a process of cleavage. Okay, so this particular process cannot be found in any other organism types except for animals. So when we speak of cleavage, that's when the now zygote, the fertilized egg, starts dividing into ever smaller cells to form a hollow ball of cells we call the blastula. Okay? And the hollow here in the center is called the blastocell, which is filled with a liquid. Okay? And eventually this blastula is gonna fold onto itself to form layers of tissues. So this is now a process called gastrulation to produce a gastrula. And a fully formed gastrula will have 3 distinct layers of tissues, embryonic tissues in it. So first we have the outer layer, the ectoderm, and then we have the middle layer, the mesoderm, and the lining in the middle, in this case represented by the yellow layer of cells, we call the endoderm, and surrounding or being surrounded by the endoderm is a pouch, a blind pouch we call the archenteron. And then the archenteron opens to the outside via the blastopore. Okay? So the blastopore either becomes the mouth or the anus of the animal depending on what type of animal we're talking about. So this type of development cannot be found in any other types of organisms except for animals. Once the development in the womb is done, or in the egg is done, or once the animal hatches, okay, there are certain stages to the development which is marked by what we call as metamorphosis. So the animal becomes a juvenile animal through metamorphosis, starting from, say, a larva, okay? And there are even animals that undergo complete metamorphosis wherein the larva hides itself inside a cocoon and then it emerges later on as an adult, a fully developed adult. But there are also some animals that have the juvenile resembling an adult. So just like in humans, okay? So, but in humans, or I think a better example would be some types of insects like a grasshopper, for example. So a grasshopper doesn't undergo complete metamorphosis, but the juvenile grasshopper looks like an adult grasshopper, except that it's not yet sexually mature. Well, we can say this also for humans, such as children. Children might look like adults, miniature adults, but they are not sexually mature yet. Okay, so this is a simplified phylogenetic tree of the animals. Okay, so the animals are believed to be of a sister taxon of a type of protists we call as the choanoflagellates. Okay, choanoflagellates meaning their cells have collars shown here, and then there's a flagellum per cell. Okay, and they live in colonies as shown in this drawing. Okay, so that's our sister taxon, and we, together with sponges, are classified under kingdom Animalia. Okay, so sponges are quite different from all the other types of animals, so that within kingdom Animalia, the sponges are our sister taxon. Okay, the sponges also have retained that same collared cell as the original choanoflagellate shown here. But the cells in the sponges specialize more on their colonial lifestyle, meaning to say there are cells that work mainly on getting food for the sponge, and then there are cells that work mainly on the distribution of nutrients through the sponge's body. But still, sponges don't have true tissues. And I think I've mentioned this before, that if you actually disintegrate the cells of a sponge, they will survive. And later on, they will come back together to form a sponge. Okay, so they're quite independent. But for real animals, the sister taxon to sponges, once you actually disintegrate the cells, they will not survive anymore. Okay, so this one is a simple phylogenetic tree showing the relationship between our sister taxon, the choanoflagellates, and to our even more specific sister taxon, the sponges. So animals are also unique in that they have a molecule called a cadherin, which allows their cells to actually recognize their own cells and stick together. Okay? So shown here are the differences in the structures of the cadherin molecules found on the cell surfaces of the cells of animals. So let's talk now about the history, the prehistory of animals. So the diversity of animals that we know of right now is actually just a fraction of the total diversity that have ever since colonized and died here on Earth. In fact, I think you're very much aware that there were actually more animals that have now become extinct than those that are still extant nowadays. So this diversity and number of animals actually was believed to have exploded during the Cambrian era. Okay, we call this the Cambrian explosion. Okay, and it was also during this era wherein the bilateral animals have actually evolved from their much simpler radially symmetrical ancestors, like the jellyfishes. Okay, so these bilateral animals have a complete digestive tract. They have a one-way digestive system with a mouth and then separate anus. And then they can also move forward because they now have distinct heads and tails. So we know that it was during this time when the explosion of the diversity of animals happened because of the multitude of fossils found in layers of rocks that are dated to be as old as the Cambrian era. Okay? So in fact, scientists have made models of giant arthropod-like creatures that would have been really scary if they were alive today, like giant centipedes and giant millipedes, which you can ride on, giant dragonflies. Those were all believed to have existed during this Cambrian era. So, in terms of body plan, we may also classify animals into two. Okay? One would be a radially symmetrical animal, okay, wherein there is no distinct head nor tail, and the animal's digestive tract opens just on one entrance and exit. So the mouth also serves as the anus. So an example of this would be the jellyfishes, okay, or more generally, phylum Cnidaria and Ctenophora, okay, or the comb jellies. And then we also have the bilateral animals, wherein their bodies may be divided into two mirror images of each other, okay. So this time around, they have a distinct head and a distinct tail, and their digestive tract starts from the mouth and ends at the anus, okay. So bilateral animals are actually more active because they can now sort of have a direction when they move. Like when they move, they can move forward or backward. While radial animals like the sea anemone that we have here usually are sessile or sessile. They can either just free float in the oceans and then wait for chance to get or to touch a particular food matter through their tentacles. But they cannot move in, say, directed movements like how bilateral animals can, okay? And also an advantage of bilateral symmetry is there's also a head, as I mentioned a while ago, where it houses the brain and all the senses. So all the senses are here in the head. So when the animal moves, it's really the head that is directed first. towards the motion because it's used for sensing. While for radially symmetrical animals, their senses are distributed throughout the periphery of their radial symmetry. Okay, so for bilateral animals, we have terms such as dorsal, that's the back. So if this is our animal here, okay, so we have our head. So we have the dorsal part here on top, okay? And then we have the ventral here at the bottom, and the front is now the anterior portion, and the back is the posterior portion, okay? So these anatomical terms are only applicable for animals with bilateral symmetry, like humans. Okay, so in terms of the layering of the tissues, animals also have their own unique characteristics not found in plants or any other organisms. Well, except for sponges, because sponges don't have true tissues. As I mentioned, if you break apart their cells will survive and later on will come again to become a new sponge. Okay? But true animals, diploblastic or triploblastic, okay, meaning to say 2 layers of tissues or 3 layers of tissues, actually have these specialized layers for specific types of organs inside their bodies. Okay? So, typically when we say diploblastic animals, these are also the radially symmetrical animals, like the jellyfishes and comb jellies, like the cnidarians and the ctenophores. While triploblastic animals, which now have the mesoderm in between the ectoderm and the endoderm, are all bilaterally symmetrical animals, like us humans. We can also classify animals in terms of the presence or absence of a body cavity. Okay, we call the coelom. Okay, animals with this type of body cavity are known as coelomates. So I don't know if you're aware, but your internal organs are actually not directly attached to your skin or to your muscle layers. There is a gap between your internal organs and your muscle layers, and that gap is actually our coelom. So the coelom helps protect the internal organs from direct shock or blow from external forces. Okay? In fact, in many animals which don't have internal skeletons, like this earthworm, the coelom or the hollow between or inside the body is filled with a fluid which acts as the hydrostatic skeleton of the animal. Okay? So this is a coelom. And we can see here the 3 layers of the body of this animal: blue for ectoderm, red for mesoderm, and yellow for endoderm. And the coelom is surrounded completely by the mesoderm. Okay, so that's a true coelom. We also have animals that have body cavities, but these body cavities are not completely surrounded by the red layer. As you can see here, on the inner portion, it's the yellow layer, it's the endoderm. So this one is not a true coelom, so this is a pseudocoelom. Animals that have pseudocoeloms are known as pseudocoelomates, and an example will be roundworms, like your ascaris or yung bitok, no, the bulate. Still, we have animals with no body cavities at all, and they don't need it anymore because their bodies are so thin, okay, dorsoventrally, like flatworms or members of the phylum Platyhelminthes. So you can see here their bodies are solids and there are no body cavities within the mesoderm. Okay, another way to classify animals is through the pattern of cleavage or cell division of the zygote once it undergoes cleavage, the cleavage process. There are animals whose cleavage follows a spiral pattern. Okay? So, an example or examples of these animals are mollusks and annelids. So, like your snails, clams, and even your earthworms and leeches, they follow a spiral cleavage, which is also determinate. Determinate meaning to say that each of the cells here in this blastula is already determined to become a certain body part for the adult. So it's determinate. For us humans, we share a characteristic known as radial and indeterminate cleavage. That's also the reason why we may get a single cell from a blastula, specifically the inner cell mass, if you can remember that in our cell biology, and that inner cell mass can become a whole new organism. Okay? That's because each of those cells are indeterminate still, meaning to say they don't have a destiny to become a certain part or an organ of a body yet. Meaning to say they have the full potential to become the whole organism. So examples of animals that undergo radial and indeterminate cleavage are echinoderms, like your starfishes and sea urchins, and of course, chordates like the lancelets and the vertebrates, including humans. Okay, so this is just a detail of the formation of the coelom, especially during gastrulation. Okay, so we see here how the blastula is folded so that there is actually one entrance, okay, into the inner cavity of the gastrula, we now call as the archenteron. So the archenteron actually becomes the gut of the animal later on. Okay? And based on the animal, there are two fates of the blastopore. Okay? For animals known as protostomes, like the mollusks and annelids, okay, which also undergo spiral and determinate cleavage, their blastopore, the first pore from their gastrula, becomes their mouth. But for us humans and the echinoderms and all the other vertebrates, we are deuterostomes. Deutero meaning the other. Stome, stoma, hole. So that means our mouth comes from the other hole, not the blastopore. Instead, our blastopore, the original hole in our gastrula, when we were still gastrula, becomes our anus. Okay? And our mouth comes later on in our development. So until now, there are still much activity as to the proper classification and phylogeny of animals. So not so long ago, we just based our phylogeny of animals on their morphology, maybe even on physiology. But right now, we are very much dependent on DNA technology and then molecular biology. Okay, so we even base our classification of animals on how their Hox genes, for example, genes that work during the development of the animal, have evolved or have modified itself. Okay, because as we know, Hox genes are shared by all animals, even plants. So this is like a shared derived character. But as humans, our Hox genes is a bit different from that of, say, the fruit fly. So the Hox genes, studying the Hox genes and the sequences of the gene and the product proteins also gives us light guidance as to how to classify animals into their real phylogeny. Okay, so here are 5 important points about the relationships among living animals. Number one, all animals share a common ancestor. So the animal kingdom is monophyletic. Sponges are the sister group to all other animals, as we've seen a while ago. And all the other animals are known as eumetazoa, meaning to say true tissued animals, while sponges are actually known as the parazoa. And then number four, most animal phyla are made up of bilateral animals. Okay? There are three major clades of bilateral animals, all of which are invertebrates except for the chordata, or those with notochords and eventually vertebrates. So here's a more detailed phylogeny of the animals. Okay, so you've seen here our sister taxon is really the sponges, phylum Porifera. Okay, and everything else is eumetazoa, meaning to say true tissued animals, while the sponges are parazoans. Okay, so you notice here we belong, phylum Chordata. Everything else are without backbones or invertebrates. And you notice here how, if we just consider the invertebrates, how we form a taxon that's actually paraphyletic in nature. So this is what we mentioned way back then, that the group of invertebrates is actually not a formal group because we have missed a particular group of animals, the chordata, if we just include all invertebrates. So here are future questions with regards to the animal kingdom. Are sponges monophyletic? Are ctenophores basal metazoans? Are acyllomate flatworms basal bilaterians? Okay, when we say basal, meaning to say, are they the simplest of the metazoans or the bilaterians? So in your generation, when you become scientists, I hope some of you will be able to give light as to the answers to these questions.